can do it up here.
Good.
There we go.
Yeah, good.
So yeah, here's a few slides and then that's a laser pointer.
Okay, thank you. Sorry. Thanks, Michelle. Yeah, so good morning. Just going to give an overview of some of the molecular research we're undertaking at the State Herbarium. I'll just start by acknowledging Cor over here, who has been responsible for a lot of the hardcore methods development in the lab, and also Michelle, who, amongst her several talents, makes sure we can pay our bills, which is always really appreciated. And so I was just going to talk a little bit about so-called next generation molecular methods, and then give some examples of how we're using these methods to inform our research at the herbarium. I'll just start with... Oh, no, it's the pointer. Arrow. Sorry. So, I'll just start with this slide, which shows how the cost of DNA sequencing has fallen dramatically over the last several years. This line, Moore's law, is the halving of computer costs per annum, and is seen as a reasonable benchmark for a well-performing industry. So compared to that, sequencing costs and sequencing is just really taking off. And just to give you an example, in the present we can sequence roughly 100,000 complete human genomes for the equivalent cost of a single genome back in 2001, which is really quite astounding. Now this largely reflects the development of so-called next generation or high throughput sequencing techniques, which allow us to sequence millions of DNA fragments in a single sequencing run, compared to one sequence per reaction using the older style technologies. And needless to say, this is changing all areas of biology. Now, this is an example that's quite relevant to our research at the herbarium. In the top part of this slide shows the number of plant gene sequences generated in a single, albeit relatively large-scale sequencing study across the plant tree of life, which is approximately 2 orders of magnitude greater than the totality of gene sequences for plants presently housed in public repositories. And this provides us with an enormous resource. For example, in our bread and butter research, it helps us to develop new markers for DNA barcoding and phylogenetic studies, but it also opens a range of new opportunities. And for example, we can begin to look at evolutionary novelties in various plant groups from the perspective of gene evolution. So it's quite an exciting time. Now, molecular research at the herbarium is largely aimed at supporting its key roles, which include biosystematics, and its areas including molecular plant identification, phylogenetic systematics, and understanding how historical processes such as past climate change have influenced the patterns of diversity we observe in the present-day flora and biogeography. There's also a focus on ecological and conservation genetics, and we use molecular data, for example, to inform the management of endangered species. We work on a very diverse range of species, essentially the whole of the South Australian flora and beyond. And in many cases, our species are quite poorly known, and typically they have no little or no existing genetic resource. So often we have to start from scratch. A number of our projects are one-off or small scale and bring relatively limited funding. So I guess our challenge over the past few years has been developing simple and low-cost methods in line with some of our limitations that allow us to harness the exciting potential of new sequencing techniques. Over a number of... I've lost it. Oh dear. No, it's not coming back. Am I pressing the wrong button? No, sorry, I was pressing the wrong button. What a goose. Yeah, so over quite a number of years, amplified fragment length polymorphisms, or AFLPs, have been a go-to method for poorly known small-scale type studies. And they have a number of advantages because, for a start, it's cheap, and because they're based on restriction sites, they require no prior knowledge of the genome and can be applied to virtually any system. They generate highly variable data that provides a reasonably unbiased representation of the target genome. There's a number of drawbacks. For example, it's difficult to determine whether two fragments of approximately the same size across individuals represent the same part of the genome. That is, homology assessment is difficult. There are also a number of limitations in terms of how we can analyse presence-absence data and therefore the inferences that can be drawn. So one of the methods that we've been working on and developing as a core method is termed RAD sequencing. It starts essentially with an AFLP kind of approach, but rather than visualising fragments on a gel, we sequence the fragments, which I guess gives us the advantages of AFLPs while avoiding some of the pitfalls. And so in a typical study, we can generate up to millions of sequences per individual across hundreds of individuals, which represent potentially thousands of independent parts of the genome. And we can do this for somewhat less than $10,000, so it's a relatively cheap and very accessible and fairly powerful source of evidence. I'll just talk about a couple of studies where we've used this approach, starting with some recent and ongoing work on Carpobrotus or pigface, which is common in coastal dune communities where it's an important, plays a fairly important role as a dune stabiliser, so it's quite widely used in restoration projects. Within the Adelaide region, there's a single native species, Carpobrotus rossii, and at least one introduced species, including Carpobrotus edulis. And on the basis of a morphological study, our expert taxonomist, Helmut Tolkien, he's up the back here, suspected hybridisation was occurring, which of course has several implications for restoration projects. So we've been asked to perform a population genetic analysis to confirm the occurrence of hybridisation and also to assess the extent. Now, this is some results from one of several sites, which is reasonably representative. This is from Torrens Island. And the top part of this slide shows the output from a structure analysis, which is a software which essentially attempts to identify the number of underlying genetic groups in the data. Here, the best solution was 2. Now, each group is represented by a different colour, and each individual, I think there's 49 individuals, are represented by a horizontal bar, and the scale on the left shows the proportion of the genome of that individual belonging to each genetic cluster. So what these results show, the red cluster corresponds reasonably well with Carpobrotus rossii. The green cluster corresponds reasonably well with the introduced species Carpobrotus edulis, and we have a number of individuals which show mixed parentage, confirming the presence of hybrids. Now, the bottom part of this slide shows the output from an analysis which uses the genotype data to assign individuals to a number of predefined genealogical classes. The pie chart shows the proportions inferred for these data, and basically what it shows is that a lot of the hybrids are actually back-cross individuals, that is, they're hybridising and then they're crossing back with the parental types, which in the long run can lead to complete loss of pure parental genotypes. So what our initial results suggest is that any restoration projects using Carpobrotus need to consider control of the introduced species as well as the hybrids. Now, seagrasses. Now, seagrasses are flowering plants which have secondarily evolved to life in the sea. I've had a talk class, and so we've been involved in secondarily evolved to life in the sea. It's quite a diverse group of unrelated species, and almost without exception, the taxonomy, particularly at the species level, is difficult, and this has been a bit of a major headache for Michelle through most of her botanical career. So we've been trying to apply these new methods to try and get some resolution in seagrass taxonomy, including hologyny, which includes one or several species, depending on the taxonomy you follow, with a quite extensive Indo-West Pacific distribution. And I think our preconceived notion was that we'd find a single species, perhaps showing geographic structure throughout its range. And what we actually found, this is again a structure plot, is 3 very distinct genetic groups, along with hybrids, including all possible combinations of these 3 groups. What does it mean? That's what we got to. So in contrast to Carpobrotus, most of these hybrids appear to be early generation hybrids, so there's no evidence of back crossing. So the parental groups, if you like, appear to maintain their integrity. In terms of the distribution, you can see that there's really no geographic pattern. Am I getting confused? I was just about to. There's no pattern, and I should have practiced my time.
That's right.
There's a few more slides. Thank you.
Sorry for cutting you off, Ed. But
No, that's fine.
Thanks, Ed. Cheers. especially when it was just getting interesting.